Welcome everyone. My name is Tracy Rose and I'm Director of Patient Engagement at the Children's Tumor Foundation. We had been planning for months with Dr. Oliver Dunca and his team at the Ohio State University Wexner Medical Center to revive the NF2 gathering. We were eager to bring everyone together in person to benefit from an intimate learning experience with fun social activities and to establish and renew networking opportunities. Due to COVID, this was not realized as we had envisioned, but we hope that you enjoyed the expressive art session earlier and that you will benefit from the informative program we have designed for this evening. At this evening's meeting, you will hear from NF experts about the past, present, and future of NF2 research. Before we get started, just a couple housekeeping rules. First, for the hearing impaired, a closed caption is provided. There's a CC button at the bottom of the screen. Please click the button and you will be able to adjust the size of the captioning. If you have any questions or challenges with that, please share that through the chat function. This webinar is being recorded and will be posted online once we have had the opportunity to review and caption it properly. Everyone is muted if you're not a panelist. If you're unmuted at some point, please make sure you mute yourself after you speak. If after this webinar, you're looking for more information about CTF and the work we do, it is available at www.ctf.org. Again, welcome and thank you for joining. Thank you for helping CTF support you. This meeting was made possible with planning support from the Ohio State University Wexner Medical Center. Thank you to the amazing planning committee. The members were Dr. Oliver Dunca, Beth Miles Markley, and Stephanie Miller. With that, I would like to turn it over to our president of the Children's Tumor Foundation, Annette Backer. Thank you.
Thank you so much. First of all, I'm not going to repeat what Treysan said, but I think we're all missing each other. And let me just do a quick shout out to our friends at Ohio for revitalizing the NF2 community. So I have one quick slide that I wanted to share with you, if I can do that. Okay, so for those of you who are less familiar with the Children's Tumor Foundation, we are really focusing on building bridges within the US, building bridges now also to Europe. And I think we are doing a lot of amazing stuff, and you will hear a lot tonight of all that great stuff. And I don't want to disclose anything because our friends will be presenting these amazing things. I just wanted to quickly put the situation in perspective and saying, what is it in fact that CTF is doing and where is our added value? And in fact, one of the observations that we had is that in a normal, let's say traditional R&D ecosystem, you have the clinics, you have the academic enterprise, as I always call it, with basic translational and clinical research. You have the pharmaceutical companies in biotech, which I have depicted here as commercial. And then you have the regulators, which are the FDA and the EMA. And in a normal, let's say traditional R&D ecosystem, these stakeholders are relatively isolated on their islands, all doing great stuff, but relatively disconnected. Our dream, our passion, or whatever you want to call it, was really, is there a way that we can connect all these stakeholders with each other so we can obtain two things? First of all, we can make sure that we make new discoveries and that we keep funding research, both through the Discovery Fund and the Synodos. The difference here is that this is more a competitive, let the best one win. And this is more like an Olympic team approach. This is a problem no lab can solve on its own. So let's come together globally to fix these big problems that our patients have identified. The second thing that we're really focusing on is making sure that every discovery is maximally translated into better treatments for patients. And therefore, we have developed an R&D enabling platform, we call that, which is really a number of tools to make it very easy for pharmaceutical companies to enter into the NF space. A registry, a target scan, a data hub, a biobank, preclinical initiatives, and clinical endpoints. And way more. I just could only fit 4 bubbles here, but it's a completely integrated platform that helps us make sure that every discovery that can be translated into better treatments will be translated into better treatments. And I think a unique characteristic of this whole model that we have built is that we're not working for the patients, but we're working with the patients. And a proof of that is also tonight, the patient engagement team that is led by Trey San. Our patients are evaluating clinics, reviewing grants. They're part of ad boards on pharmaceutical companies, and they go to the regulators to make them understand what it means to live with NF. So needless to say that we are busy, but that we are extremely excited and inspired by everybody and very much looking forward to tonight. Have a wonderful evening, and let's end NF together. Back to you, Kate.
Thank you so much, Annette. Yes, we are very excited to get started. It's been really fun to see everybody checking in the chat and telling us where you're from. As we go into our first presentations tonight, I want to encourage everyone that you are more than welcome to, in the chat box, put in any questions you have about the presentations you're seeing, because we will have just a little bit of time, maybe not as much as we'd like, but a little bit of time for you guys to hear from our speakers tonight later in response to questions. So we're going to go ahead and get started. Our first presentations come from Dr. Oliver Adunka and Dr. Varun Varadarajan, who are both from The Ohio State University.
Hey, good evening. Thanks for joining virtually today, and thanks all for being here. It's a great pleasure to be able to host this meeting. We've had, as you all know, we've hosted the Ohio Gathering for years and years, and obviously this year is very different. We're just glad you were able to make it this way. And yeah, thanks for joining. Today, want to give a brief view of clinical trials in NF2. I've asked our neurotology fellow, Dr. Varadarajan, he goes by Dr. V, to help me with this presentation. So I'm going to kick it off, and he'll give you guys some overview and what kind of opportunities are out there in terms of what new drugs are out there, where those trials are. So please feel free to also use the chat, maybe to engage with others and us. And if you guys have any questions, feel free to ask them. So I'm going to maximize my screen here. So again, state of clinical trials. Again, I'm going to kick it off, and Dr. Varadarajan will go from there. Here are my disclosures. And I think an important one to keep in mind is that this presentation will include the discussion of off-label drugs. The topics that we will go through is really twofold. One is the clinical presentation, diagnosis of NF2, and really why we want to have more, newer clinical trials and drugs available for folks who have NF2, and then really emergent treatments, what is out there. So again, NF2 is due to a gene, really, that codes for Merlin and schwannoma. It's a tumor suppressor gene. It's usually inherited, but again, 50% are about inherited. The other 50% of folks with NF2 are what we call de novo mutations, where there's no family member affected. It is passed down as an autosomal dominant path of inheritance, means that every generation usually have the 50% chance of getting it, although there is some incomplete penetrance, mean that even if you have the gene, it doesn't mean that you will get the disease. Most sporadic vestibular schwannomas actually, interestingly, have the same mutation. And as we all know, the vestibular schwannoma is sort of the classic NF2 lesion that we are dealing with. Prevalence, really the numbers vary quite dramatically on where in the world you live. It's from anywhere between 1 in 25,000 to 1 in almost 100,000 folks who currently have this. The average age of diagnosis is about 25 years of age. However, that maybe even younger children. I've seen 5 and 6 year olds, but in some folks are really not diagnosed until they're in their 40s or 50s. And correspondingly, there are 2 main types, and there's several genetic alterations that has been linked to each one of these. There's the Wishart type and the Gardner types, and it really depends on the severity and the age of onset. Again, and those have been correlated with different genetic defects. The hallmark of NF2, as we all know, are vestibular schwannomas. They're also called acoustic tumors. There can be multiple other central and peripheral nervous system tumors. Again, the most common one with has been a schwannoma. If they're, again, on the acoustic nerve, they're called acoustic tumors or acoustic neuromas. Again, there are other cranial tumors, optic gliomas, spinal tumors, peripheral nerve sheath tumors, and affected individuals also have some skin changes. We call it NF2 a neuroectodermal syndrome, simply because the skin and central nervous system are really arising from the same tissues during development. So those NF2 plaques at the cafe au lait spot, those are milk coffee spots, and some folks have cutaneous schwannoma, so that's less common. Cataracts and hamartomas are also part of NF2, usually diagnosed by ophthalmologists. That's why for us it's really important to also have an ophthalmologist part of the team. Here are the diagnostic criteria for NF2. So as definitive, NF2 is really bilateral vestibular schwannomas and a first degree family relative with NF2 and unilateral schwannoma. So if you have a unilateral schwannoma and you have a first degree relative with NF2, you probably have NF2 and are less than 30 years of age. And there are some less definitive, variables here that are listed out on the slide. So those are to consider when diagnosing NF2. Obviously, there's a genetic way to diagnose NF2, and but again, the bilateral vestibular schwannomas are most commonly associated with NF2. So these are some examples. This here's the left ear. This is the right ear, and we can see nicely here the right side of the vestibular schwannoma and the left side of the vestibular schwannoma. And here we can see a little meningioma. Here's another meningioma. Those are the 2 vestibular schwannomas, and here probably a jugular foramen tumor. Here's another folks with NF2 with some other meningiomas here on the we call those convexity meningiomas. And then we have ependymomas in the spine. So those are sort of classic tumors in NF2, right? Meningiomas are the second most commonly identified lesion in NF2. They're also benign, slow-growing tumors, and about half of folks who have NF2 also have a meningioma. Some are along what we call the neural axis. This picture here is one that's what we call the petroclival meningioma. It's a little more anterior, posterior to what we sometimes see. But the meningiomas can be a little more difficult to manage, can be a little more difficult problem for us in terms of surgery. It really depends on their location and how we address these. And there are also atypical meningiomas, which can sometimes act more like more aggressive tumors, sometimes even more of a cancerous kind of tumor. Right? The vestibular schwannoma, most common one, it's benign. We may actually, in NF2, folks who have NF2, we may deal with more than one in one internal canal. So in this area, you can have a schwannoma from the classically the vestibular nerve, which is the balance nerve, but sometimes we can have schwannomas from there are 2 balance nerve from either balance nerve, and then there's the acoustic nerve, so the hearing nerve as well as the facial nerve. So it's really difficult to ascertain exactly which cranial nerve they arise from. And sporadic tumors are definitely a little different than NF2 genetic tumors in terms of their firmness and their aggressiveness. So that's certainly something to consider, that sporadic tumors behave often very differently than NF2 tumors. Right? Hearing loss, as you can imagine, is the main concern with vestibular schwannomas. Balance problems are part of it as well, and vertigo. When tumors are larger, they can push against the fifth cranial nerve. We call that the trigeminal nerve. It can cause some facial sensation, right? And ultimately, with facial weakness, those would be for very, very large tumors or multiple tumors. And interestingly, the facial nerve can also have a schwannoma. So obviously, there are concerns with very large tumors that they can be a threat to life. So a tumor this size obviously would block the outflow of cerebrospinal fluid from the brain, basically, and causing hydrocephalus. So those would be instances where we would have to act more quickly. Most commonly, and by far more common, is that this is a quality of life kind of a situation where we want to preserve function rather than emergent surgery. Again, vestibular schwannoma is about 90 to 95% bilaterally in the folks who have NF2, and the substantial variability on how these present. Most commonly, however, and that's why neurotologists are so critical at the center of this, many times vestibular schwannomas are either the first tumor, but oftentimes they're really the most significant medical issue that folks are presented with. In management of vestibular schwannomas, we have multiple tools available. In general, we can observe, which is, I would argue, most common in NF2. We can operate, we can take these out, and we can radiate them, right? And what we'd like to talk about today is, some of those clinical trials where we actually try to stop growth via, again, a non-surgical, non-radiation approach. Just a few words to each of these approaches. Observation, again, most common in patients with NF2. Many tumors show no growth up to a certain degree. However, true growth patterns are often hard to, exactly delineate, given that we're looking at two-dimensional MRIs and not the three-dimensional, nature of the tumor. It's really hard to assess that sometimes. Sometimes it's very clear. Sometimes it's a little harder, and usually you need a few MRIs over time to really get a sense of this. Surgery, there are multiple approaches. Some of these allow us to preserve hearing. So with very small tumors, one approach is really to go in and take the tumor out, or at least debulk the tumor, and not take hearing away. There's a middle fossa approach is a classic one, so that's certainly one way to do this. Radiation the goal is really to stop tumor growth. We don't like to radiate tumors where we don't have a documented growth. And I would argue, and I would add here, that for NF2, we really don't like to use radiation simply because folks who have NF2 have a tumor suppressor gene issue, and we don't like to radiate a genetically altered tumor. So it's a little, it's very controversial, not just a little controversial, I would say, but really overall, we'd like to, if possible, not radiate folks who have NF2. And then hearing rehabilitation with either, of course, trying to save hearing as much as possible, as long as possible, really, and then use either cochlear implants or brainstem implants. Our preference are cochlear implants, since brainstem implants, as many of you know, do not work as well as cochlear implants, but sometimes it's the only option that we have available. A multidisciplinary team and approach is very important, including a neurotologist, a neurosurgeon, neuro-oncology, ophthalmology, maybe a radiation oncologist, again, that's controversial, geneticist, and of course audiology to really cater to hearing needs of affected individuals. And treatment priorities are really we want to make sure, you know, obviously if there's a life-threatening issue such as hydrocephalus, you know, really fluid buildup in the brain, those kind of things, that we address these properly together with neurosurgeon. But overall CNS function is really critical, right? And then facial nerve, yeah, unfortunately, it's common that the facial nerve cannot be preserved, but it's really our priority to preserve the facial nerve, right? Hearing, and I would sometimes argue that hearing is in NF2 is almost above the facial nerve. It's really you know, a priority for us to hearing for the longest period of time. Balance usually falls by the wayside, and a lot of affected folks have a real balance problem. In addition, there are some spinal tumors that certainly do not help. General health, mental health, those are all our priorities, I would say, in folks who have NF2. I think the next topic or next section is drug-based treatment, and I'll pass this on to Dr. Varadarajan. Again, thank you so much for joining us today. I hope everyone's doing well. Obviously, different times, and especially different times. If you're a chronic patient, you have to come into the hospital. So I appreciate you joining us this way, and I'll be happy to take any questions. I'm sure Dr. Varadarajan will at the end of this talk. And I hope you have a good meeting.
Hello, everyone. My name is Varun Varadarajan, and I'm the Neurotology and Skull-Based Surgery Fellow at The Ohio State University and Nationwide Children's. I work very closely with Dr. Adanka, and I wanted to thank you all for allowing me to join you today. I'm very excited to share with you what we learned so far about drug-based treatments for neurofibromatosis type 2. Now, drug-based treatments are also known as biologic therapy for neurofibromatosis. They're an ongoing area of study in clinical trials. There are many potential mechanisms or targets for these biologic treatments, and no drug has been shown to be 100% effective. Just because a patient or a trial has shown that there are very good outcomes, they may not be right for you, and they may not be right for every patient. Several drugs are in various stages of clinical trials of tumor suppression and the inhibition of a tumor's growth. Now I'm going to share a number of these drugs for you, and if it's an active or ongoing trial, I will provide the clinicaltrials.gov identifier number or the NCT number on the trial. And if after this presentation you want to reach out to us and find out what a particular NCT number was, we're happy to share that information with you so you can learn more about it. Here I've summarized the timeline for drug development, and this is a good overview so you can understand the different phases of drug development. And many of these trials are in phase 2, which means that the researchers are trying to assess the efficacy of a certain drug. Now, before a drug even gets to that stage, and this is not only true for drugs, it's for any new medical treatment, but it starts off with preclinical or animal testing, in which the efficacy, the toxicity, and the earliest signs of safety that we can gauge from a drug are investigated. Then it proceeds to clinical testing. Phase 1 of clinical testing primarily looks at the safety in both the pharmacokinetics and pharmacodynamics. Now, pharmacokinetics are another term for describing what your body does to metabolize the drug, how quickly the drug is metabolized in the body, or in short, what the body does to the drug. Pharmacodynamics are what the drug does to the body, and it really sort of tells you the interaction between your body and its metabolism and the drug under investigation. Then once phase 1 is complete, it oftentimes takes 1 to 2 years, the average duration for many studies. It goes on to phase 2. And phase 2 studies are investigating the signs of efficacy. Once phase 2 studies are completed, the studies move on to phase 3 clinical trials. Phase 3 trials compare the effectiveness of a new agent to the standard treatment that's in existence. And once phase 3 trials are complete, the FDA reviews the data and determines if the drug is going to be released to the public. Now, like I mentioned before, there are many potential mechanisms or sites of action for biologic medications. And now, this is a very complicated flow chart that I'm not going to go into in detail, but it sort of summarizes a handful of cellular mechanisms for growth and proliferation of cells. Now, as you heard about, Merlin is the protein or molecule that is transcribed by the NF2 gene. And inactivation results in abnormal cellular mechanisms that result in cell proliferation and tumor growth. Now, many of the medications that are under investigation, like I've listed here, can target various different cellular mechanisms and enzymes that can inhibit tumor growth and ultimately stop growth or even potentially even shrink the tumor. Now, these are some of the drugs that I'll be discussing today, but I sort of wanted to show you how there are many potential sites of action that are under investigation, and the researchers are currently even discovering more potential sites of activation. Today, I'm going to discuss the following medications, and I'm not going to go into too much detail. I just want you to be aware of some of the trials that are currently underway. And like I said, I'll provide the NCT number, and at the end, I will provide some links as well for you to learn more about these trials if you're interested in learning more. Now, some of these are completed trials, and some of these are active trials that are recruiting. And I'll mention that if it's an active and recruiting trial, that it's something that you'd be able to look into to see if you would potentially be a candidate for the study. I also, at the end, will list a few medications that were considered to have poor responses, and I have that on a separate slide for under poor response trials. The first medication I'll discuss is Bevacizumab, or Avastin. Avastin is an anti-VEGF monoclonal antibody, and VEGF is vascular endothelial growth factor. It's another cellular mechanism or molecule that helps angiogenesis, or the growth of new blood vessels for a tumor. And now this antibody helps prevent the blood vessel growth, ultimately to stop the tumor from getting a blood supply. Its administration is an IV or a port, and there have been a couple of phase 2 trials that are completed. And actually, one of the bigger trials showed a 41% hearing improvement in adults with vestibular schwannomas and a 23% tumor response as well. Another trial showed a similar rate of hearing improvement and a 43% tumor response on imaging. That being said, Bevacizumab, or Avastin, its efficacy has been shown for schwannomas only, and there are risks to this medication as well as every biologic that we'll be discussing today. But some of the well-known risks are high blood pressure, kidney failure, severe bleeding, nausea, fatigue, headaches, skin rash, and especially impaired wound healing. And the reason this is relevant is because a lot of patients who are candidates for Bevacizumab or enroll in a clinical trial may be considering surgery for their vestibular schwannoma at some point. That being said, you'll need to talk to your physician to see when you should stop your Avastin or your Bevacizumab before undergoing surgery, and when you can restart your medication as well after surgery is complete. Avastin is approved treatment in some countries, although currently it's not approved by the FDA for vestibular schwannoma treatment. The next medication I'll discuss is Axitinib. This is a VEGF receptor and TKI inhibitor. I know this is a similar target mechanism as Avastin and follows the same vascular growth pathway. It's a pill, actually. It's an oral medication, and there's an active trial at NYU, phase 2 trial that they're not currently recruiting. And the study will test if Axitinib will shrink vestibular schwannomas. And if this proves to be promising, then oral administration could actually be more practical than Bevacizumab, which is, like I said, is an IV medication. Axitinib has also been described for both breast and kidney cancer. The next medication I'll discuss is Selumetinib, or AZD6244, and this targets the MAP kinase pathway, and it's a MEK inhibitor. This is also an oral medication, and there is a phase 2 trial that's currently recruiting at Cincinnati Children's, so that's not too far from us. This study will test whether or not Selumetinib will affect NF2 tumors, and this includes schwannomas, meningiomas, ependymomas, or gliomas, and they'll typically pick a target tumor and see its response on the tumor growth. They'll also record things like hearing, daily activities, and the overall physical and emotional health of the patient. The next medication is Icotinib, and this is an EGFR receptor, or epidermal growth factor receptor inhibitor. The administration is oral, it's also a pill. There's actually a phase 2 trial in China that's examining or investigating this drug in Beijing. Now, unfortunately, the status is unknown about this trial, but it is listed in the clinicaltrials.gov website. This study will evaluate tumor volume for NF2 related tumors, hearing ability, as well as quality of life. The next medication I have listed here is Bigatinib, and this is an EGFR or ERB-B2 inhibitor. This targets the RTK pathway, and I know I'm going to be mentioning these pathways and mechanisms, but this is more so for your reference, and you don't really necessarily need to understand the exact cellular mechanism of how these medications work. But this is also a pill, and there was a phase 2 trial that was recently completed, and also at NYU. And this trial showed a 31% hearing improvement and a 24% target tumor response. Major side effects are actually uncommon, but some common side effects are memory loss, acne, light sensitivity, as well as liver damage. And actually the medication has been approved for breast cancer, and so hopefully there'll be more information coming on this medication. The next medication I'm going to discuss here is something that we're all very excited about. This is called Brigatinib, and this is an ALK inhibitor. It is a pill, and there's a big multi-center phase 2 trial underway. The main sponsor is Massachusetts General Hospital, and it's active and recruiting. It's also known as the Intuit NF2 trial, and that stands for Innovative Trial for Understanding the Impact of Targeted Therapies in NF2. Most trials are investigating a drug's effect on a target tumor. This trial is unique that it monitors all NF2 tumor types. And so if a patient has several tumors, the study is going to look at the effect of the medication on the growth of all of these tumors. And now we have to obviously have the Children's Tumor Foundation, the Synodos for NF2 effort to thank for this trial. The next medication is AR42. This is a histone deacetylase inhibitor. This is an oral medication. A phase 1 trial has been complete, and this was the first human trial for this medication, and it targets NF2 tumors. Thus far, it's been very well tolerated. The drug does reach the patient's schwannomas, and the preliminary results show that there is some anti-tumor activity with AR42. The next medication I'll discuss is AZD 2014, or Visducertib. This is an mTOR pathway inhibitor. This is also a pill, and it's in a phase 2 trial currently, which is active but not recruiting. The results of this trial are pending. The sponsor for the trial is Massachusetts General Hospital. And this medication is also being studied to determine if the drug will shrink or slow the growth of recurrent meningiomas in non-NF2 patients. So we look forward to the results of this trial. The next trial actually takes a look at FAK inhibitor, that's the GSK medication, and an HH inhibitor, which is Bismuthigin. It is an oral medication. Both of them are oral medications, and a phase 2 trial has actually been suspended. The sponsor was the Alliance for Clinical Trials in Oncology, and the intention was to study NF2 patients with growing meningiomas. There are also several medications that were considered to have poor responses. That includes Erlotinib. This is an EGFR inhibitor, and it's been shown to be not effective for hearing or tumor response in patients with fast-growing tumors. RAD-001, or everolimus, has also been shown to be not effective. Nilotinib, that trial ended as it resulted in death. But there's also several other medications, including Dasatinib and aspirin, that have been shown to have a poor response. The future directions are actually very exciting, and gene therapy has promise as far as treatment for NF2 and several other medical conditions. Gene therapy refers to the introduction of DNA into a patient to treat a genetic disease. It may replace a mutated gene, it may inactivate a mutated gene, or it can modify an existing mutated gene as well. The NF2 gene is relatively small, which actually makes it feasible to be carried by a viral vector. That puts NF2 patients as a great population that may be benefited by gene therapy in the future. And there's more information about this at the NF2biosolutions.org website. That website actually summarizes 4 laboratories that are working on gene therapy. And one of these laboratories is actually Dr. Meyer's lab right at Nationwide Children's down the street from us. The Meyer lab is actually investigating NF2 gene therapy to be able to insert the functioning NF2 gene into mutant cells to be able to halt tumor growth. I have listed here several other websites that may be beneficial to you. The clinicaltrials.gov website, obviously, you can learn about any of these trials. the NF2is.org. This is a great website for NF2 information and services. Obviously, the Children's Tumor Foundation.org. Thank you so much for hosting us today. The Advocare website actually has information on clinical trials as well. And lastly, CenterWatch.com has clinical trial information listed on their website as well. If you're interested in a clinical trial, make sure you speak to your clinician about enrollment, and they can use any of these resources to find out who to contact and to see if you're a potential candidate for recruitment into a clinical trial. I'd like to thank you all for your time today. Thank you for including me. Thank you, Dr. Dunca and Ms. Keltz, for inviting us to speak today. We're happy to answer any questions that you may have. Feel free to reach out with any other questions as well.
Wonderful. Thank you so much to both Dr. V and Dr. Dunca. We're now going to move on to one more presentation before we have our first live Q&A session. And this presentation is brought to you by Dr. Longsheng Chang, who is a renowned expert in the field of NF2. So I'm looking forward to this.
Hi. My name is Longsheng Chang. I am an investigator at Nationwide Children's Hospital and the Ohio State University. First, I would like to thank the CTF for inviting me to speak to you about the work of Synodal for NF2 on the identification of Brigatinib as a potential treatment for NF2-associated meningioma and schwannoma. Synodal for NF2 Consortium represents a multi-institutional collaboration consisting of investigators from 8 academic institutions around the world, the NIH NCATS, and the SAGE BioNetworks. Along with the patient representative Tracy and Salvo from the CTF. The team utilized a systems biology approach to identify the effective drug for further clinical development. In another word, we use the matched NF2 expressing and the NF2 deficient cell to identify drug or drug combinations that selectively killed NF2 deficient cells. Using a robotic tool shown in here, Mark Further from NIH screened a library of about 2,000 compounds, which consist of drugs that are either FDA approved or are in active preclinical and clinical developments. Drug and drug combinations that are potently inhibiting NF2 deficient cells were selected for further animal evaluations. This slide shows you the top 3 synergistic drug combinations for NF2-related meningioma and schwannoma models. Interestingly, ALK1 and anaplastic lymphoma kinase inhibitors appear to be effective in both meningioma and schwannoma cell models. In addition, ALK1 in combination with the AKT inhibitor NK2206 showed a synergistic growth inhibition in meningioma. And similarly, ALK1 in combination with the tyrosine kinase inhibitor dasatinib also showed a synergistic growth inhibition in schwannoma cell models. At the time of the discovery, Area Pharmaceutical, which developed ALK1, have developed a second-generation inhibitor called Brigatinib with much better drug-like properties. Therefore, the team decided to further evaluate Brigatinib instead of ALK1. To determine whether Brigatinib is as effective as ALK1, the MGH team led by Vijayas showed that indeed Brigatinib potently inhibits meningioma growth, and it can synergize with the AKT inhibitor MK2206 for further growth inhibitions. Similarly, the Florida team led by Christina also found that Brigatinib effectively inhibits schwannoma cell growth, and it can synergize with dasatinib for growth inhibitions. Using an orthotopic NF2 deficient meningioma model, we showed that Brigatinib can cause tumor shrinkage as compared to vehicle-treated control, which steadily grows over time. In addition, combination of Brigatinib with the AKT inhibitor MK2206, a larger tumor shrinkage and a faster tumor shrinkage was also observed. As NF2 patients may require a long-term treatment and may need to take a break from treatment due to side effects, we showed that meningioma will regrow upon cessation of Brigatinib treatment. However, retreatment with Brigatinib can further shrink tumors. This result suggests that a patient may be able to take a drug holiday if it is needed during Brigatinib treatment. The Indiana team, led by Dr. Claps, also showed that Brigatinib inhibits schwannoma growth, as shown in here that a Brigatinib-treated tumor is smaller and can improve the hearings in genetic engineered NF2 mouse models. Following our findings, Brigatinib received FDA approval for the treatment of ALK-positive lung cancers, and the drug brand name is called Alunbrig. In conclusion, Brigatinib has potent anti-tumor activity in NF2 deficient tumors. Based on these findings, CTF have collaborated with the drug maker Takeda, which now owns Area Pharmaceuticals, initiated a multi-center clinical trial called Intuit NF2, which you have just heard from the previous speakers. And this trial is led by Dr. Plotkin at MGH. I would like to point out that Brigatinib is not the only potent drug for NF2 that the Shinoda for NF2 team discovered. The dual mTOR1/2 inhibitor called INK128, shown in the middle panel here, also appear to be effective in suppressing meningioma growth compared to vehicle-treated control. In addition, among the top compounds or compound combinations identified from the library screening, there are several other potent inhibitors that need to be further investigated. These include histone deacetylase inhibitor, proteasome inhibitor, kinase inhibitors, tubulin inhibitors, and topoisomerase inhibitors. In addition, we have identified two natural compounds that potently inhibit both NF1 and NF2 tumor growth. So it is anticipated that more effective drugs will be discovered in the future. So please stay tuned and never give up. So I would like to thank the Children's Tumor Foundation, the Galloway families, and the U.S. Department of Defense for supporting our research. Thank you very much.
Excellent. Thank you so much to all of our presenters this evening. So I've been watching the chat box and trying to sort of keep up with some of the really great questions that we're seeing. Before we start in with that, I do want to encourage if our presenters who are here with us this evening, if you can turn on your video and unmute yourselves, I will present some of the questions I've been asked, and feel free to answer whichever ones are relevant to your work and that you want to jump in on. And I just want to encourage everyone that if there are questions that we do not have time to respond to tonight, do not worry. We keep an eye on all those questions, we record them, and we're happy to reach out to you afterwards. I'll also include my contact information in there so that you can get a hold of me following our presentations tonight. Okay, so the first question I'm going to ask came in through chat and said, has there been any research looking specifically at the impact of some of these drugs, such as Brigatinib, on vestibular schwannomas that have already been treated with radiation, particularly if growth has actually been stopped or inhibited as a result of radiation? Or are these clinical trials, are people considered ineligible if they've previously had radiation treatment?
So yeah, generally speaking, you wouldn't be considered eligible if you have a stable tumor that's not growing, right? The only exception maybe if there's progressive hearing deterioration over the the course of time, that would maybe be an exception to that rule. But generally speaking, these trials are for growing tumors and specifically to stop growth.
Mm-hmm, mm-hmm. Yes, thank you, Dr. Adanta. Another question coming from chat tonight was, how exactly, well, maybe not exactly, but how are drugs chosen to move into that human clinical trial phase? Maybe an example of kind of how we've done that in the past, but it seems like there are so many options and so many things that have been tried. So maybe just for the non-medical, non-research person to help understand how that process works.
I guess that's a great question for Longsheng. I mean, he's been at the forefront of this. Longsheng, do you want to tackle this one?
Okay, yeah, try to give a 2 cents kind of comments there. Basically, usually we want to make sure first is that the drug is safe and show efficacy there. So that's why we go through multiple different kind of models. And so even in the cell cultures, we do in even patient tumor cells, as well as some available cell line or genetic engineered cell line. And then we move into the animal if it's really efficacious there. And so in the animal, we want to make sure, again, animal is well tolerated. And sometimes we even do the so-called toxicity study. So we want to make sure that in animal, we do not see any organ toxicity. And then, of course, most importantly is also showing that it will be able to shrink tumor or kill tumor cells. And then from there, sometimes if it's more serious kind of conditions, we might even take it to a larger animal. So for example, we might even do a dog test or monkey test before we move into the humans. And so with all of those things there, then if everything is go well, and the drug is potent, and then we will move into the clinical trials.
Excellent, thank you.
Yeah, quite a complex process, and some of us have seen this now with, of course, what's in the news, the COVID vaccine, right? I mean, it's not a simple process, even for the accelerated one that's going on right now, and we're all observing. So Longsheng has been really at the front of this. So we rely on people like him.
Absolutely, thank you. It is definitely an effort of teamwork. Absolutely. Okay, so in the interest of time,
this is also why those take a few years to develop for the whole process, and then get to these stages. So
yeah, absolutely. Thank you so much to both of you. So we're going to move to our next presentation. Keep the questions coming into chat. Our presenters will stay with us tonight, and we will have one more Q&A towards the end. Okay, so next we're going to hear from Dr. Marco Giovannini. Unfortunately, he's not able to join us live tonight, but please feel free to continue to ask questions in chat related to the presentation you're about to see, because we do have some other experts here tonight who may be able to speak to some of those questions. So I'm looking forward to hear what he has to say. He's going to be talking to us tonight specifically about gene therapy, which I've already seen questions about. So let's get started.
Good evening, everybody. I am Marco Giovannini, faculty in the Department of Head and Neck Surgery at UCLA and co-director of the UCLA Multidisciplinary Neurofibromatosis Clinic. I will give you a short introduction on the potential of gene replacement therapy for neurofibromatosis type 2. What are the challenges to therapeutic intervention in NF2 in general? First of all, the benign nature of the tumors precludes the use of strategies that target rapidly dividing metabolically active cells, such as those of malignant tumors or cancers. Next is the reduced therapeutic window for treatment that these benign tumors provide. And then the question of exploiting unique features of NF2 tumor cells to target the broad consequences of NF2 protein loss. A very important question before we even think of gene replacement therapy as an option is, would NF2 re-expression, even if successfully delivered, stop or reverse tumor growth? In order to do so, we need to identify tumor-specific therapeutic targets, strategies for delivering therapeutics, and validate NF2 gene replacement as a viable therapeutic strategy. If you look at similar situation where inactivation of a tumor suppressor gene is the initiating, the causative event in tumorigenesis, we immediately see, when we do a bibliography search, is that there are very few examples of gene replacement strategies in conditions similar to NF1, NF2, and schwannomatosis. So one is the approach of restoring p53 function in tumors. And the idea was, the proof of concept was provided by the lab of Tyler Jacks in Boston. They showed that restoration of p53 function in mice leads to tumor regression in vivo. And there are a couple of therapeutic approaches ongoing, but there is very little information out there about both of them. The first one is from China, Gendicin, which consists on intratumoral injection of adenoviral vector expressing the p53 gene. It has been cleared by China FDA in 2003 for head and neck squamous cell carcinoma, suggesting positive results and safety. Since then, it has been tested in a number of clinical trials for other cancers. However, no information is available about the submission of clinical data for approval from the US FDA. The other one is a similar adenoviral p53 vector for this time systemic intravenous delivery. And again, no FDA approval yet. So in summary, adenoviral p53 gene therapy seems to be well tolerated and feasible, with promising anti-tumor effects in some cases, in some tumor types. However, the overall clinical efficacy is not conclusive, and no FDA approval yet. Another example is re-expression of the TSC1 gene, the tuberous sclerosis gene, in a genetically engineered mouse model. This was done by the group of Sandra Brakefield at MGH. And what was interesting here is that they showed that, again, an adenoviral vector expressing the TSC1 gene was able to increase survival time of mice, of tuberous sclerosis mice, by at least 3 to 13-fold, and that increase in survival was accompanied by normalization of sizes of ventricle and neural cell bodies in the brain, which are hallmarks of the disease. And overall, this was well tolerated. So what are the challenges for gene replacement therapy for NF2? First is the vector selector, using a viral such as the AAV. But if that's the case, which serotype would be most efficacious in targeting Schwann cells and arachnoid cells that are the cells of origins of schwannomas and meningioma? The other vector selection option could be non-viral, for example, exploiting characteristic of NF2 deficient cells, such as micropinocytosis proficiency, as suggested by Andy McClatchy lab. The other challenge is optimizing transduction efficiency and selectivity. For example, intratumoral delivery versus systemic delivery, intravenous or intra-CSF, cerebrospinal fluid, and what are the NF2 expression level and expression pattern of mosaicism that are necessary or sufficient for efficacy. Another challenge is, of course, what is the safety of acute re-expression of NF2? Of course, if we inject viral vector, for example, expressing NF2, would this sudden re-expression have some deleterious effect? So we need to test all these variables before we can even think of adopting the strategy in vivo in patients. So our take on this, our idea was to genetically evaluate NF2 gene replacement as a therapeutic strategy in vivo by generating a new strain of mice in which NF2 re-expression can be induced by injection of adenoviral protein called FLIP-E. So injection of the adenovirus expressing this protein in the NF2 deficient mice which develop NF2 tumors would eventually shrink or at least stop the proliferation of those tumors. So what we know so far is that at least in vitro, the trick seems to work, as in cells originated from those mice, expression of the FLIP-E efficiently results in re-expression of Merlin, and cells in which Merlin has been, or the NF2 protein, has been re-expressed, stop growing. This experiment was performed by, in the lab of Andy McClatchy in Boston, and she kindly allowed me to show this unpublished result. Next, we are working on what is the best delivery route of viral vectors or macrotherapeutics to NF2 tumors. Jeremy Vitt in my lab found that injection in the CSF at the level of the cisterna magna, which is just below the cerebellum, results in efficient diffusion of the viral solution targeting the skull base, meninges and cranial nerve, cerebellum, olfactory bulb, and as well as along the spinal cord, DRGs, dorsal root ganglia, and spinal root and nerves, which are often tumoral in NF2 patients. So in summary, we think our hypothesis is that viral and macrotherapeutic delivery are viable options for NF2 gene replacement in NF2 mutant tumors. And these approaches initiate a new pipeline for preclinical studies in NF2 schwannomatosis, specifically using the new NF2 mouse model of schwannoma and meningioma with a reversible allele that we develop. Of course, advanced testing of these approaches for safety and efficacy will require scaling up in larger animal models, such as pigs, before translating into clinical trials in people. I just want to mention my team, Dr. Jeremy Vitt, our collaborator, long-standing collaborator, Dr. Andy McClatchy, Christine Chasson-McKenzie, Dr. NF2, the co-director of the UCLA NF clinic, Dr. Michel Calamaridis in Paris for the meningioma model, and our collaborators at other institutions. Thank you, good night, and unfortunately I will not be able to participate to the Q&A session because I'm in Europe, so the time zone does not allow me to be there with you, but please do not hesitate to email me if any question. Good night.
Wonderful. Thank you so much. I know he's not here, but I hope everybody really enjoyed that presentation. I'm really enjoying seeing all of you interact with each other in the chat as well. That's always been such a strength of this meeting, is the time together and just the way that benefits the whole NF2 community. And so it's been really great to see that happening even virtually. So next we're going to hear from, well, it's Dr. La Rosa, but if you work at CTF, we know him fondly as Salvo. Salvo is going to be speaking tonight with a few different people that I will let him introduce and sort of explain. But following his presentation and the discussions you're about to see, we will have one final Q&A session. So please keep the questions coming in. And now I'll hand things over to Salvo.
Hi, I cannot switch on my camera, so please, control room, if you can help me. In the meantime, I will start sharing my screen. Okay, I hope you can hear me.
Yes, we're hearing you just fine. Thank you.
And can you see my slides, Kel?
Yes, yep, we can see everything.
Okay, so let me just switch this. Okay, now you should see my full presentation. Okay, so thank you for being here and allow me to present today about the NF2 accelerator. And I like a lot this this picture about this comic strip about the accelerator by Moxie and Spark that explains to Maria how the accelerator works. It really is about speed and accelerate science. So let's dive into it. First of all, as an outline of today, I will talk briefly about investments in NF2 and schwannomatosis that CTF has done in the past years, just to put this into perspective of where we are now. And then I will dive into the NF2 accelerator. Special guest for the NF2 accelerator part is Michael Wooten, CEO of NF2 Therapeutics, in charge of our gene therapy approach program for NF2. And he will join me with some comments explaining where we are with this study. Then we'll talk about the nonprofit marketplace, a new business opportunity that CTF has put in place for companies to get new drugs into the clinic. And then at the end, we'll have a special guest discussion with Edith Garrett and John Morris to understand from them as patients and caregivers what they think about the NF2 accelerator, why there are two accelerators of this program. Okay, so as a start, you saw from Annette earlier that CTF business model is really about connecting, catalyzing, and having patients at the center. But what I want to focus your attention is that, in this bit of catalyzing and investing in NF2 and schwannomatosis, CTF has been very active with these two models mainly. The Discovery Fund, which is a fund that funds the best and boldest from bench to bedside, and is very competitive grant program. Is one of those that they send us the application, we select the best, and we fund it. And then the other one is a team science approach with Synodus that we heard from Long Cheng about the Synodus NF2, where we actually built a preclinical pipeline that allowed us to identify Brigatinib, and it ran between 2014 and 2017. And now the Synodus for schwannomatosis that analyzed largest number of tumors and is wrapping up right now with a publication that is coming up very soon, as being already approved. But the most interesting part that I want to drive you on this slide is that since 2006, which is the last 15 years that CTF has been operating, 33% of CTF funds, this is the entire budget that every year we give to research or we allocate to research, has been dedicated to NF2 and schwannomatosis. So it's not that the incidence of NF2 and schwannomatosis is less important. It's as important as NF1. And sometimes we find compelling studies that we fund just because the science is at the level that we're required for funding. So this huge investments of about 15 million in the last 15 years has really brought us to the cusp of discovering and bringing the first drugs to patients. Okay, so now the NF2 Accelerator is a program that basically it was activated in 2019 after we completed the Synodus NF2 as the follow-up study that we thought was most appropriate. Is basically formed in its part by many different, but three or four different parts. Is a 2.36 million investment program to expand the drug pipeline and accelerate treatments. So it's a 3 years undertaking. We are already halfway through, and the 3 or 4 parts are basically expanding the clinical drug pipeline for NF2, and I will show you 2 different modalities that are connected, 1 and 2. Then the second part that we thought was critical to include in the accelerator was improving drug selection, because sometimes we've been successful and we've seen a very good results in mice and animal models, but this didn't really translate into effective treatments in patients in clinical trials. And then the other part is the development of gene therapy option that address the underlying genetic cause of NF2, and we'll hear from Michael there. So first part is really the basket platform trial for NF2. And you heard already about Intuit in the list of clinical trials that are now ongoing. And the drug that is the first drug that we have on this platform trial is Brigatinib by Takeda. It's an ALK inhibitor, but the group believes, and I saw questions on the chat, that it's not by its main target ALK that this drug works, because ALK actually is not expressed in NF2 cells. So it's one of the many off-target effects that this drug has. So this is a multi-kinase inhibitor and has a lot, hundreds of targets that inhibits a different potency. So teams already identified that target and the publication is ongoing. But if you think about this drug and the Intuit platform, basically how this works is that, once you have patients that enter the trial monitors all of the different tumor types that NF2 has. And depending on what tumor type best respond to the drug treatment after an evaluation, if there is a radiographic response or stable disease, patients stay on trial and receive the drug treatment. It keeps on being on the trial. Or if there is progressive disease, meaning the trial doesn't work, it's put off protocol in the natural history arm and actually is back to the allocations. So it may change the arm or may change drug. So the status of this Takeda Brigatinib trial is that, of course, the main PI is Dr. Procking at Massachusetts General. We have 5 participating sites, and 1 site at MGH is already open and 6 patients are already receiving the medication. Importantly, Takeda has a big role here because the trial is co-funded by the NF2 Accelerator and CTF and Takeda Pharmaceutical. Takeda is putting in $1 million, and CTF is putting through the NF2 accelerator 300K. Now think about how this can really accelerate drug testing. And I would like to think about this trial, because this is the real benefit of having a platform trial, is like, you know, you can basically have this as a main socket where you can plug different drugs to the same main protocol. So if you think about the first drug, Brigatinib, then, you know, the second drug, third drug, fourth drug, and fifth drug will come very easily. So we don't have to basically use a different protocol. We use the same master. We'll cut bureaucracy of trial opening and only amendments to add new drugs. So that basically is a huge, huge speed up in this process, that we will be able to plug on this trial multiple drugs and have a very coherent way to evaluate drug response and efficacy. So the end result is that in very short time, patients have access to multiple drugs and mechanism that as they become available, as this drug become available for NF2. And we are also actively talking to pharma and biotech to get their drug into this Intuit platform. What is the second level of, you know, how we actually push those drugs into clinical trials? And the other way to do it is to really fund new ideas and new drugs testing for NF2 and schwannomatosis. I think I presented the first 3 awards that we funded through the NF2 Accelerator to Vijaya Ramesh, looking for drug combination from Synodus NF2 that were as potent as Brigatinib. And then, you know, new oligo antisense that were studied by Castellano in Barcelona, and HiPoYAPTAZ inhibitors studied by Chanling He. This is a very interesting mechanism for NF2. These teams are now wrapping up their results. There's been some delays due to COVID, but we are close to, you know, seeing the first outcome from these grants. But then we also funded recently YAs, for instance, at the University of Plymouth and at Columbia University, that are looking at more like basic science into the same HIPPO pathway, looking at new inhibitors, new way to interfere here, but also new substrate, for instance, for schwannomatosis that connect to LZTR1. And very recently, we assigned other grants to Jeremy Vitte at UCLA for micropinocytosis for therapeutic delivery to cells, and to Dr. Lei Zhu at MGH for the treatment of schwannomatosis-associated pain. So far, the NF2 Accelerator has funded supported for these grants for $678,000. And we'll keep doing it. We'll keep assigning the best and brightest, promising scientists more grants so that new options will become available soon. The third part of the NF2 Accelerator, as I said before, is to have models for NF2 that will allow us to have better decision and broader accessibility. For this part, we are working with Marco Giovannini at UCLA and Wade Clapp under the GWR company, that is a non-profit CRO, to look first of all at NF2 mini-pigs model. And this study is basically monitoring if mini-pigs will develop the same feature as NF2 patients do, so that we have a close to human model that will allow for better evaluation of drugs. But also with Wade Clapp, we are trying to create mouse models that are accessible to everyone. Unfortunately, we've seen so many collaborations failing because companies, they're not able to strike a deal with an academic institutions and get access to their models and their research facilities. This has been, for many years, a problem that either delays enormously or, you know, completely the deal is not happening. So we are now collaborating with Taconic to create models that are no strings attached. And unfortunately, we had to start from scratches, create those models because there are IP and there are, you know, patents that institution that created, they still own. So in order to free them from everyone, CTF will collaborate with Dr. Wade Clapp and make this available to any for-profit, non-for-profit organization that wants to test drugs for NF2. That will open a new stream of drugs available. And last but not least, of course, we have our gene therapy for NF2. And I'm very glad we have Michael Wooden, CEO of NF2 Therapeutics with us. And Michael, I don't know if did you unmute yourself?
Yeah, I'm here. Glad to be here, Salvo.
Thank you so much, Michael. And I actually put, you know, those bullet points that highlights where we are with the project, but I would like you to comment and update everybody on where we are first, but where are we heading, right? Thank you.
Sure, absolutely. Thank you, Salvo. And, you know, first of all, I'd like to just say that we're very grateful for the support of the Children's Tumor Foundation. They have provided support to NF2 therapeutics for a long period of time. And we've been sort of the best kept secret for some time, but we've made great progress. And you know, in a very sort of clear time frame, with the support of the Children's Tumor Foundation, we'll be able to achieve proof of concept in gene therapy, or demonstrate that that it's not going to be viable. We are obviously very, very optimistic. And you know, just a quick little bit about the company before I talk about where we are. So it was founded by myself, Dr. Scott Plotkin, and Mark D'Souza. It's actually my second NF company. The three co-founders also founded a company called Inflection Therapeutics. And we have a topical MEK inhibitor in clinical trials for cutaneous neurofibromas in NF1. Currently, so very, very excited about that. So in NF2, we have developed a gene replacement therapy for NF2. And, you know, in Marco's talk, he talked about some of the strategies for developing gene replacement therapy. Our approach is an adeno-associated viral approach. So unlike the COVID-19 virus, this is a good virus. This one's our friend because it doesn't make humans, it's not associated with disease in humans. And it is a very efficient way of delivering the gene to the cells that we're targeting. So instead of the gene of a virus developing instructions to the cell to produce copies of itself, it's delivering our instructions to produce a copy of the healthy Merlin protein. And so we have developed the gene therapy, and we have recently completed a 28-day study in non-human primates. And so specifically, cynomolgus macaques are the species of monkey. And there were 6 monkeys involved in that study. And essentially, what we're looking for from that study is one, confirmation of delivery, so confirmation that we see the transgene in the cells of interest, and then also safety. So we're looking at any potential signs of toxicity within the monkey study. And the fact that it is a monkey study is very important. There is a real challenge in the field of gene therapy in translating data from cells or even small mammals to what we see in humans. And so it's very important to work in a species that is as close to humans as we can. It's also what the FDA expects in terms of the data necessary to move into clinical trials. So in the study that we've completed, we in the in-life portion of that study measure an array of different characteristics, including the behavior of the animals, the weight of the animals, a full sort of blood workup on the animals at various points across the 28 days, as well as observations like skin color, condition of fur, fecal consistency, discharge, any other potential signal of toxicity from the study. And the great news is that after the 28-day study, there were no significant signs of toxicity, which is a great sort of data set from a safety perspective. So we're very excited about that. And after that 28-day study, so on day 29, the monkeys are sacrificed, and they're sacrificed so that we can do pathology on those monkeys and look at an array of tissues, specifically Schwann cells, arachnoid cells, ependymal cells, where some of the tumors that NF patients experience occur. And there we're looking for delivery. We're confirming, are we seeing delivery in those cells? And then we're also looking at a broad array of other cells, 14 different levels of the brain, other organ tissues, and we're looking for safety. So we're looking for any evidence of toxicity. We're looking at glial cells in the brain to see if there's any change in the shape of those cells, and a broad sort of cross-section of cell types. And so we expect the data from the pathology portion of that safety study, as well as the data on biodistribution of the vector and expression of the transgene by the end of the year. So we're very excited about that. And then beyond the non-human primate study, we are also working on a mouse model of NF2. So it's a knockout mouse model in the lab of Dr. Marco Giovannini. And so we will essentially be able to see if in Dr. Giovannini's model, we're seeing regression of schwannomas in NF2 and also differential growth rates between mice that have been treated with our gene therapy and the untreated mice.
Michael, what is the time to, let's say, if everything goes according to plan, what is the time that this first dose will go into human?
So the kind of 2022, 2023 timeframe is when an initial human trial could commence. We'll be driven by the data, obviously. And so, these studies will kind of steer the course going forward, but we're optimistic, and that's our goal, is really to have a first trial by sort of late 2022 or 2023 timeframe.
And last question, maybe apart from, of course, everything has to be according to plan, what is your number one worry of, problem that you see for getting there? Is it distribution, is production, what is it?
Yeah, so I think it's really the distribution question. Are we going to see expression of the transgene in the targeted cells? And so we're going to have an answer to that pretty quickly, which is quite exciting. We just heard about a study where re-expression of healthy Merlin can rescue the phenotype. And so we're excited to see the result of that as well. But it's the delivery question is the sort of key thing that's keeping us up at night.
Great. Thank you, Michael. I think I fulfilled quite a few people with that question. We're asking about, when we'll see this gene therapy in humans. Thank you so much, and stick with us, please, because there may be some other questions at the end. I'll just keep going with the presentation.
Thank you.
So my next slide is about, very quickly, because then I want to talk to our patients and caregivers. It's about, put the things in perspective. And if you think this is a timeline of short-term, mid-term, and long-term, when I say NF2, just the NF2 accelerator is really a balanced portfolio. Think about time, this as time to bring treatments to patients. So we talked about the Intuit NF2 platform trial. If you think about this trial, is already bringing drugs to patients. So the first drug is already into the socket of this platform trial, and we are ready to plug as many as we can into the same protocol. So this is ready to go to be used, very short-term project that will allow drugs that are ready to go into the clinic to move on. And then we'll learn from Michael that the gene therapy for NF2 is really not far from there. And it's a big bet, because either we hit jackpot or we fail. And unfortunately, if we fail, we need to go back to the drawing board. And that will require more time, of course, to think about how we need to supply to that change. But things will move quickly, and we'll have an answer very quickly. Hopefully, finger crossed, everybody here is a fan of NF2 therapeutics and gene therapy. So we want to basically this to succeed. And then if you think about the medium term, how do you fill that gap? And what is the NF2 accelerator doing? Is investing in new ideas. Because today we have a knowledge about what we can plug into that platform trial, but we don't know if there are new ways to stop tumors to grow. And that's why we need to go back to basic biology, and we need to increase our funding and understanding of basic mechanism by which these tumors are proliferating and acting. So that's where our investments are so important. And then, plug into this new animal models that we are trying to make available to everybody. And that will actually free a lot of momentum for other companies to bring their own drugs and test them in relevant animal model, have a very quick decision on, is this drug making sense for NF2? It doesn't. For schwannomatosis, it does. We don't know, but it will allow us in the short to medium term to have a stream of drugs coming from, hopefully coming from the industry. And this is powered by the nonprofit marketplace, a new business model that I talked to you before. And I want just to explain briefly. What we created is a marketplace partners where 3 companies, the Children's Tumor Foundation, the Milken Institute, and Cure Search for Children's Cancer, they are putting together this marketplace that you see here in the middle. So how this works is basically composed of experts in the field of drug development. That is not per se only CTF or the Milken or Cure Search, but there are external people that have extensive experience in bringing those assets to the clinic, where they're screening, they're identifying assets, they're externalizing those assets and frameworks, and they're making matchmaking to develop new partner. So how this works, basically there is a partner that is pharma and biotech company that will bring their assets. So we're talking to them to free these assets, put it in the nonprofit marketplace, and then after an assessment of this drug into those models that I was talking before and those results that I was talking before, we'll go back to the partners and create an accelerator for those assets, or create companies of those single assets so that they will be able to develop this drug. The good news is that these models is quite interesting for investors, as the first venture partners is already on board, and BridgeBio is willing to put their own investing money into this model, sustaining the development of this drug, and of course benefiting from the outcome and potential revenue that this drug will generate. So there you have. Thank you so much. I really appreciate the opportunity to talk, but now I would love to have Edith Garrett and John Morris with us. And you guys, can you just say something? Just
Here's Salvo. I'm here. John's here.
And is Edith there as well? Okay, she will join, I hope, or let me know if she's not with us. But John, thank you so much. I know you've been involved with us so long, and you are an NF2 accelerator. So can you tell us why you got involved in these initiatives?
Sure, I can, Salvo. Thanks for having me. So I've been involved with CTF for probably approaching 10 years now. My youngest son was diagnosed with NF2 when he was 10 years old. And he's doing quite well, but as all of you know, it's a lifelong situation. And my wife and I decided that if there's a way we can help, this is where we were going to put our time and money. And so we've done exactly that, and I'm proud to be an NF2 accelerator and hope we can encourage a lot more people to do so as well. So
tell us a little bit more about your role. I mean, what do you do? I mean, as an accelerator, as an NF2?
Well, so we formed a committee with the help of our friends at CTF, and along with my co-lead, RB Harrison, we are trying to do two things. We're trying to get the word out about the NF2 Accelerator program so that we can raise money for some of the things that Salvo and the other experts here just talked about. And we're trying to build awareness for NF2 and the great things that CTF and the NF2 Accelerator program are doing in the marketplace to try and put an end to NF2.
Great. And is Edith with
Hi,
us?
Salvo, this is Albert in the control room. Edith is online. You may ask her questions.
Okay. So yeah, I would love to hear from Edith about why you're actually involved with the NF2 Accelerator.
Okay, can you hear me okay?
Yes.
Okay, great. So actually I was thinking about this question, and I remember about a year ago, I went to another CTF event. It was a small gathering, and it was at that event that I was able to speak to someone about, being involved, this, that, and the other. And I let her know, I said, I want to get involved. I'm a patient, I want to be involved in this community, but I didn't know how. And it's like working full time, I just didn't know how to manage it. And she kind of smiled. She was like, oh, don't worry. We have plenty of people who work, help us out and work full time. I was like, okay. So this opportunity came up about 3 months after, and she contacted me. And honestly, I'm thrilled. It's been a great experience.
So why do you think others should join? I mean, why is this program exciting to you?
Right. So as a patient, I sat here and I listened, and things I've known, things I'm learning, and I think, holy smokes, this is fantastic. We broadening the drug pipeline and gearing these research studies towards NF2, and then ultimately gene therapy. Oh my God, this is amazing. I can get behind this initiative, and I want to get behind it. So for me, it's meant a lot. It's meant a lot. And then for other patients, I think about, CTF has the line, I know a fighter, right?
Yes.
So I think, yeah, I know some fighters, but we got to do this together. We got to fight it together. So much better. This is not something one person can do. We got to do it as a community. Let's make it happen.
And John, why are you excited about this? Why are you excited about this program? John.
Why am I excited? Oh my gosh, because I'm a patient. I live with NF2. I know. So part of it is, I think that the drugs and broadening the drugs, it's the possibility of, what am I trying to say? The possibility of improving somebody's quality of life, my own, somebody else's, before we get to gene therapy. Let's get to gene therapy, let's get to a cure, but before that, let's improve our quality of life, you know? Well, that's me, I don't know.
Salvo, Edith said it all. I agree completely. It's just so exciting to see all these things coming.
Perfect. Thank you so much, guys. Then it's back to Kate.
Thank you.
Excellent. Thank you both for being with us tonight. Thank you to everyone actually who is here with us tonight. I just want to make a note that we are running a little over on time, so I thank everybody for their patience and their continued willingness to be here tonight. We are going to have to forego our last question and answer just in mindfulness of the time. But again, as I've said in chat, if you have questions that were not addressed tonight, please don't hesitate to reach out to us. My email is, I'll put it again one more time over in chat because it's easier to get it that way. And I'm happy to hear from anybody that was here tonight with any questions. So I just want to hand things back over to our president, Annette, just for a brief word from her.
So first of all, well, I have one thing. Albert, can you just close the screen 1 second? Because I just got something in here that I want to share with all of you. So look what I just got. Just while we were here. Our Lule got a cover article on the Journal of Clinical Investigation about schwannoma biology. So I just wanted to share that with you guys because I think this is closing a super exciting evening. Who would have thought a few years ago that we would be on the cover of a journal so important as a Journal of Clinical Investigation? And that is because of all your awareness work. So I just wanted to do a big shout out to everybody here and not here. Your work is paying off. So Albert, if you can quickly show the word cloud, because I think these days we are not in an NF evolution anymore. We are in an NF revolution. And I just put some words on together with Kate and Tresan. And this is the word cloud. There is clinical trials, there are drugs, there are companies, there is new initiatives, there is gene therapy, there are so many, there is patient-reported outcomes, there are patients. And with that, I really would love to introduce our NF2 Accelerator ambassador, Ian Desmond. And I really wanted to thank everybody for your support, your enthusiasm, your awareness, because as Edith said, together we can end NF.
Hi, I'm Ian Desmond, professional baseball player with the Colorado Rockies and longtime friend of the Children's Tumor Foundation. I couldn't be more excited to be the honorary chair of the NF2 Accelerator initiative. I've been able to build a relationship with a friend of mine named Ethan Brown, who has NF2. We established our relationship some years ago and have grown since to almost daily communication through text. I've been able to be a part of Ethan's life and his journey with neurofibromatosis, and to see the impact that it's played on him and his family has really shown me a different side of life that I didn't know about prior to meeting Ethan, through perseverance, courage, family, really. And I think not only just his direct family, but the Children's Tumor Foundation family. So to be able to interact with not only Ethan, but families all across the country that are dealing with neurofibromatosis, it is clear to me that the Children's Tumor Foundation is the strongest advocate for ending neurofibromatosis. And I couldn't be happier to be part of their Accelerator program, which is going to shine a light on neurofibromatosis 2, which is what my friend Ethan has. It makes it feel so much more personal to me because my relationship with Ethan, but I can't imagine the benefit or the peace of mind that it gives somebody like Ethan, who knows that there's an organization like the Children's Tumor Foundation who is setting aside specific resources and time and energy to focus directly on what he's going through. I think it's one thing to know that the Children's Tumor Foundation is there in the whole, but to know that there's a team specifically designed to focus on NF2 would allow me to sleep a lot better. I can't imagine how much better Ethan and his family are going to sleep knowing that there's such a great team behind them. I ask that you join us and consider becoming an accelerator yourself. Thank you so much for your time. I appreciate it.
Wonderful. Thank you again to everyone for joining us here tonight. As everyone has said, it is absolutely a work of a brilliant and dedicated team, scientists, doctors, researchers, and most of all, those who are living with NF2. Thank you again to the Ohio State University for helping us put this together. This is one of our best turnouts for an NF2 specific gathering, and we are thrilled that you were all here tonight. And as everyone said, together we will be able to see the end of NF2. Have a great night, everyone.